Hello, my name is Emily Marshall. Here I have my Create It Safe presentation for research I helped perform in the fall semester of 2020. The title of this project is In Vitro Bactericidal Effects of 415 nanometer blue light on Pseudomonas aeruginosa 19660. Introduction and purpose. Phototherapy or light therapy can be used to help treat many medical conditions that affect the skin. Pseudomonas aeruginosa is a gram-negative bacilli bacterium. This bacterium is an environmental species and is naturally occurring in water and soil. An opportunistic pathogen, Pseudomonas aeruginosa, is a common cause of nosocomial infections. Pseudomonas aeruginosa can also be found in moist areas of clinical settings such as sinks and respiratory equipment. Objectives. This study is based on the use of blue light therapy to treat burn wound infections. Pseudomonas aeruginosa was tested on brain-heart infusion auger and treated with 415 nanometer blue light to measure the bactericidal effects of light exposure. Methods. 50 microliters of Pseudomonas aeruginosa stock, ATCC 19660, was inoculated to 5 milliliters of brain-heart infusion broth and incubated at 37 degrees Celsius for 18 to 24 hours. The Pseudomonas aeruginosa was then washed and resuspended in phosphate buffered saline. This was achieved by centrifuging the broth at 4,500 revolutions per minute for 10 minutes. The supernatant was removed using sterile pipettes and replaced with 5 milliliters of phosphate buffered saline. The solution was centrifuged again at the same speed and time, decanted and replaced with phosphate buffered saline. This solution was resuspended using a vortex. 3 milliliters of the Pseudomonas aeruginosa solution was pipetted into a small petri dish to be treated with 415 nanometer blue light. The light therapy doses range from 0 joules per centimeter squared to 60 joules per centimeter squared. Following the blue light exposure, 40 microliter aliquots of the treated Pseudomonas aeruginosa solution were transferred to brain-heart infusion auger plates. These plates were incubated at 37 degrees Celsius for 18 to 24 hours. The Pseudomonas aeruginosa growth was then diluted. This was achieved by making a 0.5 McFarland standard using phosphate buffered saline and treated Pseudomonas aeruginosa colony growth. The 0.5 McFarland solution was diluted to 1 to 10,000 using a serial dilution. 10 microliter aliquots of the diluted solution were inoculated to new brain-heart infusion auger plates and incubated at 37 degrees Celsius for 18 to 24 hours. Colony counts were performed following incubation. Findings. 415 nanometer blue light treatment inhibited in vitro growth of Pseudomonas aeruginosa on brain-heart infusion auger. The highest dose of light had the greatest effect on bacterial growth. Here you can see a graph demonstrating the effect of the dosage of light therapy on the growth of Pseudomonas aeruginosa. We had 3 successful rounds of light treatment completed through to colony count. We also recorded our data from the control run on the graph, which included no light treatment of the Pseudomonas aeruginosa stock, as well as a dose of 60 joules per centimeter squared of light treatment. We used a control group with no light treatment to establish purity of the Pseudomonas aeruginosa stock and to obtain baseline colony counts. 60 joules per centimeter squared was the highest dose that our light instrument would let us apply. As you can see, the trend line in the graph has a negative slope, indicating that the Pseudomonas aeruginosa growth decreased inversely with the dosage of blue light treatment. In other words, the Pseudomonas aeruginosa colonies that had the highest doses of light had the least amount of colony growth on average. Discussion and action plan. The results of this study are promising for potential in vivo use of blue light therapy to treat wound infections. The next step following the study will be to inoculate the Pseudomonas aeruginosa strain to laboratory rats to measure the rate of burn wound healing in live subjects. Pseudomonas aeruginosa growth was affected by all dosages of light use, but growth of the Pseudomonas aeruginosa was inhibited most by the highest doses of light. The highest dose of light used in this study was 60 joules per centimeter squared, because that is the highest dose of light treatment that is allowed for use on human subjects. The results of this study supported the data from other studies of a similar nature. Studies with similar subjects of experimentation investigated different factors that may affect the degree of bacterial inhibition, such as rate of delivery of light and the wavelength of light used. Other frequencies of light that have been researched range from 405 nanometers to 880 nanometers. Previous studies also tested other clinically significant bacterial species. Staphylococcus aureus, another common cause of nosocomial infections, was one species that was tested in previous studies. Propionibacterium acnes and Helicobacter pylori are two bacterial species that are not associated with clinical settings, but were also tested using light therapy. Acknowledgements. This research was funded with a grant by the name of the use of photobiomodulation to treat infected wounds through the Arkansas Biosciences Institute. Much thanks to Zach McLean and Carly Farmer for their support and assistance in this study. Thank you also to Claude Rector, the instructor mentor over this project. Thank you for listening.